Hello, everyone. My name is Dallin DeGraff-Reed. I guess I'm just going to go over a few of these problems here. I can figure out how to share my screen. Here we go. I think. Oh, that was weird. All right, hopefully you can see my screen. If not, I'll repeat the questions anyway. Hopefully I'm doing all this right. All right, so for the group questions, the first one is, suppose you perform the following cross, capital A, lowercase a, capital B, lowercase b, capital C, lowercase c, uppercase D, capital E, lowercase e, with, I'm just going to go ahead and say heterozygous A, heterozygous B, heterozygous C, heterozygous D, and recessive E. All right, hopefully you get the picture there. What proportion of the offspring, or I guess assume all of these genes undergo independent assortment. A, what proportion of the offspring is expected to have the heterozygous A, uppercase B, lowercase c, lowercase c, dominant D, and lowercase e. What is expected to have, what proportion is expected to have each genotype? And the answer that I got for that was 0.0078. And the way that I got that was 1/2 times 1/4 times 1/4 times 1/2 times 1/2. If you do the Punnett square for each of those alleles, those are the fractions that you're supposed to get, I guess. And then when you times all those fractions out, you get that proportion. I'm just going to assume that you can see my screen so that I don't have to keep saying all of these lowercase and uppercase letters. All right, so for B, what proportion of the offspring is expected to have the expected to have the following genotype? I'll just say that. So for part B, I have 1/4 times 1/4 times 1/4 times 1/2 times 1/2 equals 1/256, or 0.0039. Part C, what proportion of the offspring is expected to have the following genotype? All right. I have that there is a 0 proportion there. Not sure if that is right, but it's asking if you can have a recessive D gene, and that's not possible when you have a homozygous dominant D with a heterozygous D. You're always going to have a dominant trait there. All right, part D. What proportion of the offspring is expected to have the following genotype? I have 1/2 times 1/2 times 1/2 times 1/2 times 1/2. And that gives me 1/32, or 0.031. Hopefully I did some of those right. Fill in the blanks. Paired homologous chromosomes are sometimes called bivalents or tetrads. B, the structure that connects a centromere to the microtubules of the meiotic spindle is called the kinetochore. C, a diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell in prophase 1 has 8 sister chromatids, and while a fruit fly cell in metaphase 2 has 4 sister chromatids. Part D. blank is a type of genetic cross that can determine if a purple flowered pea plant has the dominant or homozygous PP or the heterozygous PP genotype. And that is a test cross. You test cross that phenotype with a known genotype, which in that case could only be recessive PP. And the next generation should tell you what the parents were. Part E, a non-functional allele is sometimes called a null allele, while a partially functional allele is called a hypomorphic. I had to look that up. Hopefully that's right. Mendel's law of individual assortment involves two traits, while Mendel's law of segregation.
Is there a marker back there behind you? Nope.
Not that I see, no. Sorry.
Sorry.
Sorry about that. I can't remember where it was. Mendel's law of individual assortment involves two traits, while Mendel's law of segregation involves a single trait. Suppose an organism has the genotype, has the following genotype. Assume that the alleles assort independently. Diagram all possible homologous chromosomes arrangements in the organism during metaphase 1. Be sure to indicate which alleles are found on each pair of the homologous chromosomes. All right, in this case, there are numerous varieties. I have capital T, capital R, capital Y can pair with another capital T, capital R, capital Y. It can also pair with a capital T, lowercase r, uppercase Y. You can also pair with capital T, capital R, lowercase y, capital T, lowercase r, and lowercase y. We could also have capital T, lowercase r, capital Y pair with capital T, capital R, lowercase y, capital T, capital R, or capital T, lowercase r, and capital Y could also pair with capital T, lowercase r, lowercase y, and lastly, capital T, capital R, lowercase y could pair with capital T, lowercase r, lowercase y. That's the best that I understand it anyway. Indicate all of the possible gamete genotypes that can be produced in this organism at the conclusion of meiosis II. So in this case, the chromosomes are going to be by themselves in a haploid cell. So this is the way I understand it. You could have capital T, capital R, capital Y. You could have capital T, lowercase r, capital Y. You could have capital T, capital R, lowercase y, or capital T, lowercase r, lowercase y. And we may have multiples of that since there are two alleles of the capital T. I don't know if that makes sense. Part C, how many sister chromatids are found per cell in this organism during metaphase 1? I have 6 chromatids. Part D, how many chromosomes are found per cell in this organism during metaphase 2? I have 3 chromosomes. A horse has 32 pairs of chromosomes. How many chromosomes are present per horse cell at the conclusion of meiosis I? Since there are 32 pairs, I think we can safely assume that there are 64 chromosomes at the conclusion of meiosis I. Part B, how many tetrads are present per horse cell during prophase 1 of meiosis. There should be 32 tetrads. I'm assuming if all of those are paired up and if we're counting all of those pairs. How many sister chromatids are present in a horse cell during prophase 1 of meiosis? Again, that should be 64. D, how many sister chromatids are present per horse cell during meiosis, during metaphase 2 of meiosis? I have 32 as being the answer. How many bivalents are present per horse cell during anaphase 2 of meiosis? Because this is the second phase of meiosis, I think we can safely assume that there will be 0 since they will no longer be paired up. Consider the following. Question 5. Consider the following tetrahybrid cross in pea plants. It gives a genotype and tells me exactly what those mean. Assume that all the alleles of the 4 genes sort independently. A. What proportion of the offspring will be tall, round, green, and terminal? And so if we multiply all of the proportions, all of the probability of getting tall with round with green and with terminal, we end up getting 3/4 times 3/4 times 1/2 times 1/2, which gives me 9/64 or 0.14. So a proportion of 0.14 will be round, green, and terminal. Part B, what proportion of the offspring plant has the blank genotype? All right, so the way I understand it, we have 1/4 times 1/2 times 1/2 times 1/2 equals 0.03. Part C, what proportion of the offspring will be heterozygous for all 4 genes? In this case, it's 1/2 times 1/2 times 1/2 times 1/2, which will give me 0.06. What proportion of the offspring will be dwarf, wrinkled, yellow, and axial? So dwarfed is homozygous recessive, as is wrinkled. Yellow has to include the heterozygous dominant yellow or the homozygous dominant, which that is 1/2, and axial is the same situation. It's also 1/2. So 1/4 times 1/4 times 1/2 times 1/2 equals 0.016. I'm hoping some of this was right, and I hope that this video is helpful. Thanks for watching.